All right, everyone, and welcome back to what will be part 3 of our discussion here in the nervous system. Remember that your next exam will only cover the muscles and the nerves, so chapters 10 and chapter 12, so there won't be anything else on there aside from that. Where we last left off is that we talked about what is a action potential and how we have depolarization that takes us from a negative number to a positive number, plus 30 to be exact, and the events that happen in depolarization where sodium ion channels will open up. And how do those channels open up? It can be a chemical stimulus, it can be a mechanical stimulus, it can be an electrical stimulus, but for some reason or another, those receptors are triggered, those channels are triggered to open up, allow sodium to come in, and it takes us from -70, which is resting membrane potential, to +30, generates an action potential, also known as depolarization. We take away those negative and positive aspects, and it's just both positive on the inside and outside of the cell. And then what goes up must come down. And repolarization, where at this point the sodium channels close, so no more sodium is allowed to get into the cell, but now potassium channels will open up, potassium will be kicked out, and we kick out potassium, those positively charged potassium ions, until we get back to a negative number. Once we get to -70 millivolts, we start to close those potassium channels, but we inadvertently kick out a little bit more potassium than we need to kick out. So we get down to -90, and then to regain our resting potential back at -70 millivolts, we incorporate the use of the sodium-potassium pump, which requires energy, or ATP. So we spent a great deal of time talking about the importance of the electrochemical gradient between sodium and potassium in our last discussion, and the events that happen in an action potential. And we even talked about different types of refractory periods. Absolute refractory, where absolutely no contraction can take place or no action potential can take place. And then relative refractory, where we can have some sort of action potential take place, but it's going to be relative to the size of the stimulus. So what we're going to do today is that we're going to move forward a little bit, and we'll talk about the effect of axon diameter on speed. We'll talk about different types of synapses. We've kind of hit them already when we talked about the different sorts of receptors we have between chemical, mechanically gated receptors, or gated channels, and voltage-gated channels. So we'll talk about the different types of synapses, and specifically, since we know about this synapse so much, we'll talk about cholinergic synapses, and then we'll have a little bit of a discussion on neurotransmitters towards the end. So for axon speed and diameter, the diameter is proportional to the speed. The ion movement is going to be related to how concentrated the cytoplasm is, and how big or wide across that axon is. The wider it is, the faster that action potential speed will be. So because the axon is so large, it's less likely to have a lot of resistance. And remember, the type of resistance we talked about a lot is myelination. So large axons tend to have lower resistance, and as a result, they're able to propagate or send that action potential very quickly along the length of the axon. So the question you're probably asking yourself is, wouldn't it be beneficial for just all axons to be very large? Well, if that were to take place, if all the axons were very large diameter axons, then that information that's receiving all of the information from about the outside of the body or the inside of the body, when it comes into the spinal cord, your spinal cord would have to be about the width of a garbage can, because in order to process all of that information. So not all information needs to be processed as quickly as some other information does. So larger axons tend to be on those sensory nerves that are responsible for relaying very, very important information. So information such as pain is something that's very important, that needs to be sent quickly to the central nervous system to make the appropriate response. So there's 3 different types of synapses that we're going to talk about. First type is an electrical synapse. Electrical synapses are locked together at a gap junction. So these types of synapses have a tendency to touch one another. So with electrical synapses, they're held together by what we call connexins, and those proteins of the gap junctions will allow for those sodium ions or potassium ions to freely go from one cell to the next. So it allows, because a gap junction is really just a tube of proteins, a tube that's made out of proteins, will allow these ions to pass between the cells. So you get this continuous local current and a continuous action potential movement. Places that we'd find these electrical synapses, many parts of the brain. and that allows for you to process information in your brain very quickly because the neurons are physically touching one another so that they can very quickly process information and send and receive messages to fellow neurons and to integrate that information into appropriate response. So think about when you're trying to read. When you're first learning to read, you need to have a relationship go to one part of your brain to figure out, is this a letter or a number? Once you figure that out, you have to figure out exactly what letter it is that we're looking at. Once you figure that out, you need to figure out what sound it makes. So notice how just from reading the word R, like are found in areas of the brain, you have to go to several different parts of your brain to pull that information. So to make that process happen more efficiently, it makes sense that these neurons are touching one another. So they get a chance to touch one another to pass this information along very quickly. So we find them in the brain, the eye, and also in your ciliary ganglia. The most abundant type of synapse you have are chemical synapses. In chemical synapses, there's going to be a chemical that's released from the presynaptic cell, and that chemical then will cause the postsynaptic cells to make some sort of response. Typically, the chemical that's released is what we call a neurotransmitter, and we'll talk about neurotransmitters at the end of the notes today. Most of synapses found between neurons and all other synapses between neurons and other cells typically are chemical synapses. A good feature of chemical synapses is that unlike electrical synapses, the cells are not in direct contact with one another. In fact, they have a little space between them we call the synaptic cleft. Now, the chemicals that are most likely responsible for sending and receiving messages are what we call neurotransmitters. Neurotransmitters are just chemicals that will be released from neurons or can be released from neurons and received by other neurons or received by other cells that will cause the receiving cell to make some changes or maybe not make changes, depending on what class of neurotransmitter is received. The one class of neurotransmitter that we've spent so much time talking about are those neurotransmitters that act in an excitatory role. They're going to cause depolarization of the postsynaptic membrane, or the cell that receives this neurotransmitter is going to go from a negative number to a positive number, aka exhibit an action potential. So excitatory neurotransmitters will promote action potentials. The excitatory neurotransmitter that we've spent a lot of time talking about has been acetylcholine, because we've talked about how when acetylcholine is released, it binds to the receptors of the motor end plate, allowing for those sodium ion channels to open up. When sodium comes in, that takes us from a negative number to a positive number, causes depolarization of the cell or an action potential. Now, there are classes of neurotransmitters that can do the exact opposite. Instead of causing depolarization to take place, they will cause hyperpolarization. So it will take us from a negative 70 millivolts to something like maybe a negative 90 millivolts, because it will cause us to get even more negative and suppresses the ability for an action potential to take place. We understand that for action potentials to take place, sodium ion channels open up and sodium comes into the cell. And we also understand for repolarization to take place, going from positive to back down to negative, that we close those sodium ion channels, we open up the potassium ones, and we kick out potassium. So for inhibitory neurotransmitters, they would trigger those potassium channels to open up and remove any of that potassium until we get to a much negative number. All of that is done in effect to suppress that cell from doing something. So it's acting as an inhibitor to the cell that's receiving this message. So the way that that neurotransmitter is received does not depend on the neurotransmitter itself. So even though we talked about how neurotransmitters can fit into these two classes, that they can be inhibitory, bless you, or excitatory neurotransmitters, sometimes a neurotransmitter can behave as both, as in the case of acetylcholine. Acetylcholine is just the messenger. How you receive that message is completely independent of the messenger itself. And a silly example I like to use is that if your best friend tells you that your significant other is cheating on you, she's just the messenger. He or she is just the messenger. They didn't cause the cheating to take place. They weren't wanting it to happen, and hopefully they're not the person they're cheating with. They're just the messenger. Now, how you receive that message, they're a butthole anyway, so I really don't care, or I'll kill them. That's completely dependent on you, the receptor. Neurotransmitters are the same way. They're just sending a message. How that cell, that postsynaptic cell, receives that message, that's dependent upon the receptor. So the effect of the neurotransmitter on the postsynaptic membrane depends on the receptor, not on the neurotransmitters, because acetylcholine, for what we have learned in the neuromuscular junction, acts as an excitatory neurotransmitter usually. It's going to promote action potentials. However, in the cardiovascular system, in the heart, it inhibits cardiac neuromuscular junction. So when acetylcholine is released at the heart muscle, it actually causes hyperpolarization to take place. It's going to suppress action potentials. So depending on what that postsynaptic cell is, acetylcholine can be excitatory, cause action potentials, or it can be inhibitory, as in the case at the cardiac neuromuscular junction. So it's not acetylcholine that's changing, it's just the messenger. How you receive that message, or how the postsynaptic cell receives that message, depends on what kind of receptors are found on the surface of the cell. So at a chemical synapse, and the chemical synapse that we are talking about is a cholerogenic synapse. It is a cholerogenic synapse because we are dealing with acetylcholine, so anything that would be in the category of acetylcholine for neurotransmitters, they're all going to kind of behave the same way. So I do want you to remember that the specific type of chemical synapse that we're talking about is a cholerogenic synapse. You have different types of synapses that are called adrenic synapses that will the binding of adrenaline or norepinephrine or epinephrine binding to those cells, the receptors on those cells can cause different types of effects, usually excitatory effects to take place. So this particular example of a synapse we're talking about involves acetylcholine. So since it involves acetylcholine, it's called a cholerogenic synapse. So we have the action potential arrives at the synaptic knob or the synaptic terminal of that knob, and we have acetylcholine that's been arranged in these vesicles that's ready to be released. And notice we have this space between the presynaptic cell and the postsynaptic cell that we call the synaptic cleft. In order to release the acetylcholine, we have to have an influx of calcium. Now, none of this should be new so far because we talked about this in the neuromuscular junction. Calcium comes into the presynaptic cell, aka the cell sending the message, and when calcium comes in, then that triggers the release of acetylcholine. Acetylcholine is then going to bind to the receptors on the postsynaptic cell or the cell receiving the message. So once acetylcholine binds to those receptors, it's going to open those channels. When it opens the channels, it allows for sodium to come in. Because this allowed for sodium to come into the cell, depolarization took place. So in this particular type of cholerogenic synapse, acetylcholine is acting as an excitatory neurotransmitter. If it were the case that there was a potassium ions were being kicked out, then we would consider that an inhibitory cholerogenic synapse, but that's not the case here. We have excitatory because sodium ions are coming in. When sodium ions come in your minds, you should be thinking, all right, we're fixing to generate an action potential. So now when we're no longer needing the sodium ions to come in, we have already had the action potential generated in that postsynaptic cell. Keep in mind that a postsynaptic cell could be a muscle, like we found in the neuromuscular junction, or it can be another neuron, or it can be a gland. So whoever that postsynaptic cell is can change, can be anything receiving the message. We've just talked about how the person or the cell type receiving the message has been a muscle. So once we're done, we have our enzyme, in this case acetylcholine esterase, will break down acetylcholine into its two different parts, and it will be retaken up by that presynaptic cell. So your body, as Fiona Apple has always said, is an extraordinary machine. It's not going to throw away anything that it can possibly find a use for. So when acetylcholine esterase breaks down acetylcholine into its acetyl group and its choline group, those two parts can be reabsorbed by the presynaptic cell and then go back to the cell body of the neuron that sent the message originally to recycle more acetylcholine. Sometimes you have what's known as synaptic fatigue, and that's when we can't recycle, in this case acetylcholine, that neurotransmitter fast enough to meet the demands of the stimuli. So you have another stimulus that comes along, and you want to keep sending this message and sending this message and sending this message, but you're unable to do so effectively because you haven't put together your acetylcholine. You haven't recycled it fast enough. So at that point, the synapse is no longer going to send the message, and the postsynaptic cell won't be receiving the message until we've replenished enough of that chemical neurotransmitter, in this case acetylcholine. Now, as you may have imagined, acetylcholine is not the only neurotransmitter we have. We have several other. There are at least 50 that we know of, but there may be up to 90 neurotransmitters other than acetylcholine. Some of them are amino acids. Some of them are peptides, which are just longer versions of amino acid sequences. Some of them prostaglandins can act as a neurotransmitter. ATP, which we're usually used to seeing as just the energy currency of the cell, can act as a neurotransmitter. And some dissolved gases, like nitrous oxide, can act as a neurotransmitter as well. Now, the only ones that we're going to talk about are kind of what I consider the rock star neurotransmitters. So we're not going to learn about all 50 of them. There's only about 4 of them that we're going to talk about. One we've already discussed at length is acetylcholine. Another neurotransmitter that we'll discuss is norepinephrine. Now, if acetylcholine is released from a special types of chemical synapse that we call a cholinergic synapse, norepinephrine is released at a special chemical synapse called an adrenic synapse. And that's very important, so I'm going to highlight that because I may ask you questions on what type of chemical synapse is norepinephrine released from. Norepinephrine usually has a very excitatory and a depolarizing effect, so it's an excitatory neurotransmitter. We find it widely distributed in the brain and definitely in parts of the autonomic nervous system. Dopamine is also a central nervous system neurotransmitter, which means that we don't really find it in the peripheral nervous system, so brain and spinal cord are kind of where it hangs out. Dopamine can be excitatory or it can be inhibitory. Unlike norepinephrine, which is excitatory and depolarizing or action potential generating type of neurotransmitter, which makes sense because if you think about norepinephrine or epinephrine, you think about things, pupils dilating and quick things happening, that fight or flight response sort of thing happening there. But for dopamine, it could be excitatory, it could be inhibitory. So we see dopamine receptors are going to be excited in cocaine use. So cocaine use is going to stimulate those dopamine receptors, and that causes them to, the receptors are more excitatory in that fashion there. And when you have prolonged cocaine use, what happens is that your body no longer becomes used to the dopamine that you make, and then your body stops making it as much as it would be. So to feel any levels of kind of the same sort of responses that you'd get from dopamine, you have to take more and more, higher and higher amounts of cocaine in order to stimulate those same types of high, in order to feel normal. When it's necessary for your body to become dependent on a drug to feel normal, that is one of the biggest hallmarks of addiction. You now become physically addicted to this as well as emotionally addicted to this drug. Serotonin, like dopamine, is also a central nervous system neurotransmitter. The job of serotonin is that it affects attention and emotional states. A lot of antidepressants work are serotonin reuptake inhibitors. A lot of like Prozac and drugs of that nature, antidepressants will work by kind of tinkering with those serotonin levels. Gamma aminobutyric acid, or GABA, is an inhibitory neurotransmitter also found in the central nervous system along with serotonin and dopamine. But unlike serotonin and dopamine, we don't really know a whole lot about it other than that it allows for hyperpolarization to take place, or it's an inhibitory neurotransmitter. It keeps a cell from doing anything, from whatever the message that's sent is don't do anything. It's not, we don't really understand the purpose behind it other than that it typically works in conjunction with other neurotransmitters. So as we talked about with cocaine, many different drugs affect the nervous system by stimulating those receptors that would respond to, such as the receptor for dopamine. Because it's an artificial response, or this response is to an artificial substance that your body normally doesn't make, many different drugs can have a complex effects on your perception, motor control, and your emotional states. Opioids are a class of what we consider neuromodulators, not necessarily neurotransmitters. Big job of opioids are to be your organic pain reliever. Opioids are going to bind to the same receptors as opium and morphine. And once again, just as we saw with cocaine, that cocaine binds to those same receptors as dopamine, that when you take heroin or any other types of morphine or oxycodone, any of those types of morphine derivative drugs, they act as pain relievers, and they're going to have a very analgesic effect to you. You have your natural, you are your own anesthesiologist. You have analgesic components, these neuromodulators that help you to deal with pain. For instance, if you ever birthed a child naturally, yes, it hurts, but your body does release these opioids to relieve some of that pain, especially in between those contractions. You can tell those opioids are working because you feel a little bit more calm. You kind of look forward to those breaks in between contractions. If you've ever cut your hand or injured yourself, you've probably noticed that when the injury first took place, it doesn't hurt as much as you would have imagined it to hurt. My husband had a cousin who got shot, and he got shot in his shoulder, and he said when it happened, he was bleeding, and he knew he'd been shot, and it was like, okay, this sucks, but he didn't really feel a whole lot of pain. Well, that's because his opioids, these neuromodulators were acting as pain relievers. Now, a couple of days later, after getting over the initial, shock of being shot and seeking medical attention, he said it hurt worse days after the incident than it did the day of. And you've probably experienced that if you've ever broken a bone or you've cut yourself, that you will experience a lower amount of pain at the time that the incident happens than a couple of days after. And that's because of these opioids. Thank heaven for opioids. We have 4 classes of them: endorphins, enkephalins, endomorphins, and dynorphins. So I want you to remember all 4 classes of those opioids. And also to remember that drugs like morphine and heroin or any of those morphine derivatives, they bind to those same receptors. So how neurotransmitters are the effects they can have is that we have what we consider a direct effect. And direct effects we talked about a lot already, also called ionotrophic effects. In direct effects, we're opening and closing ion channels based on the receptors that are stimulated by the neurotransmitter. Case in point, acetylcholine binds to receptors in the motor end plate, causes sodium ions to come in. That is a direct effect. The acetylcholine is directly affecting what that postsynaptic cell membrane is going to do by, in this case, allowing sodium ions to come in. Indirect effects are going to work through what we call a second messenger system. So indirect effects will involve G proteins, and they have enzyme complexes that will bind to these G proteins and create a second messenger system. The second messenger, or the indirect effects, there's a link between the neurotransmitter, which we would consider the first messenger, and the second messenger. A lot of times the second messenger could be something as simple as cyclic AMP. So the link between the neurotransmitter and the first messenger and the second messenger, and then we activate this enzyme called adenylate cyclase, and once that enzyme is activated, this is when we have the second messenger. Big difference between direct effects and indirect effects is that for indirect effects, it's kind of like the game Mousetrap. I don't know if you guys ever remember that, where the goal is to trap the mouse, but you have to have this really huge complex rubric on how that cage falls down on your mouse. You've got to tip over the cup of water, which then runs down the faucet and causes the beaker to fill up, which moves the weight to one side and so forth. So with indirect effects, think of it as being this complex process in order to get the cell, the postsynaptic cell, to do something. With direct effects, they're directly telling the channels to either open up or close, and we call it ionotrophic effects because the channels that are opening or closing are either the sodium channels for depolarization or those potassium channels for repolarization. So here's a pretty picture of all that happening. So let's pull out our finger here. So indirect effects via G protein, we have our neurotransmitter will bind to the receptor. Examples of indirect effects G proteins, and we're going to talk about this again next chapter in the endocrine chapter. Epinephrine, norepinephrine, dopamine, histamine, and GABA will all act this way. They have these indirect effects. So let's say that we have dopamine binds to this receptor. When it binds to that receptor, the G protein becomes activated, which then in turn activates this enzyme adenylate cyclase. Along with the help of ATP. Once that is released, we have cyclic AMP will activate the enzymes that will tell the cell to do something. So right now it says activates enzymes that change cell metabolism and activity. That just means all the crap that could be going on inside the cell. So all of those things that happen within the cell, that the things that we want the cell to do, will do now because of this second messenger. So cyclic AMP is our second messenger. Our neurotransmitters are first messenger. One of the things that it will do is that it will open or it can close that ion channel. So it does kind of two things. It opens the channel, and then it does other stuff inside the cell. And we'll talk about that other stuff that gets done inside the cell in our next chapter in the endocrine system. Indirect effects, intracellular receptors such as carbon monoxide and nitrous oxide, we find those they can bind to enzymes in brain cells. For indirect effects here, that they can just, through intracellular enzymes, they will just go through the plasma membrane, so they don't have to bind to any receptors. They'll activate those enzymes that will make the second messengers like cyclic AMP, or they can be other things that are the secondary messengers, which can still open channels or change in the cell's metabolic activity or activity. All right, so that's all we have for today. We'll finish up. We have about 6 more slides left, and we'll just finish those up on Tuesday. Don't forget your quiz. We'll cover everything that we've talked about so far in Chapter 12, and we'll cover all parts of Chapter 10. The quiz is going to be pretty straightforward questions that we'll have for the quiz. Make sure you understand the things that happen with muscle contractions, what the role of troponin is, tropomyosin, what the role is of calcium, sodium, potassium. Make sure you understand all of those things, so they'll be excellent candidates for quiz questions. And remember, it will cover Chapter 10 and almost all of Chapter 12 except for these last 7 slides. Questions, comments, or concerns? All righty. Have a great weekend, and I will see you all next week.